So this is all about DNA replication, this lesson. And the first thing we'll do is describe what she talked about at the very end of that video, and that was that the DNA replication is semi-conservative. So I've given you the term, and then I'm giving you the description of what semi-conservative is. But it's really important that you understand this process. When we make new DNA, we don't just take the old double-stranded double helix and then make a new two-stranded double helix. We're taking a piece of the old DNA and using it as a template to make the new DNA. So each new DNA molecule contains a strand of the original molecule and a new strand. Thus, each new molecule conserves half of the original. Now, if you look at the little picture clip on the side, I hope that you've seen this enough times that you're starting to recognize the structure. You've got the little circles that represent the phosphates, the little pentagon shapes that represent the sugar. You've got the nitrogen bases inside that have complementary shapes, and then they would be attached by hydrogen bonds. And then you can see right here in the middle of the picture, kind of looks like the two sides are being pulled apart. That's representing that enzyme that we'll talk about in a minute, separating the two strands. And you can see these new nucleotides kind of coming in to the middle. That's the idea of semi-conservative. The edges, the outsides, are always made up of the original DNA. And because bases are complementary, the original strand is like a template for the new complementary strand that we're going to make. Now, you already know this because we already talked about it, but this happens during the S phase of interphase. And I hope that you can see that even though this is a new topic, it relates to what we talked about in the last section as well. So it would be very reasonable for me, or the person who wrote the exam question that I put on a test, to ask you something about DNA replication and the cell cycle at the same time, because they're not mutually exclusive topics. We're talking about the same thing. Now, does anyone want to ask anything about semi-conservative DNA replication? Now, in the next few slides, you're going to see this picture over on the left. This is just a different way of representing what DNA replication looks like. The reason I like this picture is it shows me all the things I need to talk about without a ton of extra detail that's making it complicated. So if you look at the picture, there are blue strands. Those are representing strands of DNA. And then there's a bunch of what I think look like gummy candies, and they are the enzymes, all of which she talked about in that video clip that we watched. Now, things are numbered on the picture, and as we go through this, I'm going to tell you what each number represents. Now, before we can get started numbering or talking about what the numbers are on the diagram, we need to talk about two things that don't have a particular number on the diagram. The first is the origin of replication. You'll remember it from the video clip. It's a particular nucleotide sequence, so like a particular sequence of A, T, C, and G, where replication begins. Now, they mentioned this in the clip as well, but it really depends which type of cell it is for how many origins there might be. In a small circular chromosome, like what a bacterial cell would have, there's probably one. But in a linear eukaryotic chromosome, like what you have, there are probably thousands. So the idea that we're trying to convey is that we're not starting at one end of a chromosome and then just slowly moving along the entire length. Even though we're showing this happening at one spot, it's happening at a whole bunch of spots concurrently along the length of a chromosome. The other thing that you'll see and hear is the replication bubble. So the spot on the right of the picture where we've pulled the strands of DNA apart and it actually looks like a bubble, it's like a big rounded structure, that's the replication bubble. It's the separation of the two strands that creates this oval shape. It has a fork at each end. So you can see one fork right here. It's the point where the two strands start to go away from each other instead of staying along that parallel track. There would be a fork at the other end of the bubble too, you just don't see it in this picture. Now those unwound strands get pulled apart into single unpaired strands, and then we use them as models to make new strands. Now there's an arrow on this picture. It's pointing towards the left. And this gives you a clue as to the directionality of this process. As the enzymes do their job in this particular picture, DNA replication would continue along to the left on this picture. In the video, she showed it going towards the right? So that was just two different ways of representing it. But you always have to look at the replication bubble, and it'll give you a clue as to which way the process is going to move. And once we talk about the enzymes, I think you'll see that it will be easy to determine which way it's going to move. Because what you're really trying to figure out is, where is the bubble going to continue to open up? Now, if you would like to ask a question about any of those things, yeah, Caleb.
What do you mean by like a fork?
So a fork is where, see how I have those two blue lines? Those are my two strands of DNA. And see how right here, they start going in different directions? So they used to be right next to each other, parallel, and then when they open up, one kind of goes up, one goes down. Like a fork in the road, where it splits up into two different ways. That's what we mean by fork.
So just like the start of the bubble.
The start of the bubble, yeah. And there would be a fork at the other side of the bubble where it ends too. Anybody else want to ask anything there? Okay, so we're going to get started talking about the numbered pieces of this picture. So same picture, but now we're talking about the numbers. Number 1 represents helicase. Now, it's plural there because remember I said there's not just one spot that this is happening. It's happening all over the place, but we're just looking at one instance of it in this picture. So this big sort of pinky reddish oval represents helicase. Is it an oval in real life? No. We're omitting the detailed chemical structure here. But it's an enzyme that binds to DNA. It separates and unwinds the two strands of a portion or segment of the double helix. So when you are looking for helicase, it will always be at one end of the replication bubble. Whichever end it is at, that's the direction that replication is moving. So see how here, helicase is on the left side of the replication bubble? That's how I know that replication is going to continue on to the left in this picture. Now, number 2 are these blue squares, and they're all over the place. The blue squares are DNA polymerase, so that was the little worker dude from the video. He was green, he looked angry, so he was a little construction man. This is an enzyme that inserts itself into the replication bubble and adds nucleotides to create a complementary strand of DNA at the 3' end. Now you remember the first time I mentioned this 3', 5' business, I reminded you, you don't have to know the chemistry explanation for why a carbon is the 5' carbon or why it's the 3' carbon, but you do have to understand that that's how we indicate directionality in DNA. You'll notice that there are some over here and down here because of course we're making a new strand on both sides. 3 and 4 kind of go together. Number 3 is the green square. It's an enzyme called Primase. So they said this in the video, but it bears repeating. When something ends with ASE and you're in biology, it's probably an enzyme. That's how most, but not all enzyme names end, but a lot of them do. So primase is the enzyme that you need to make the primer. What's the primer? Well, the primer described below is a little chunk of RNA. But think of what the word primer means in other contexts. Have any of you ever helped paint a wall in your house? You have to put this stuff called primer on. You don't have to, but you should. It helps the paint stick to the wall. I put new drywall up in my basement, and new drywall like soaks paint in like you wouldn't believe. And without the primer, I would have needed like 15 more gallons of this paint that I was using. So the primer, when we're painting, helps things stick to wherever you're trying to paint. And it's the same idea here. The primer is a little strand of RNA that's going to help DNA polymerase figure out where it should start sticking new things. So primase is the enzyme that makes the primer. The primer itself is that little yellow bit labeled number 4. It's a different color so that you remember that it's made out of RNA, not DNA. And DNA polymerase will remove the RNA primer afterwards, like it'll replace it with DNA. Now that's numbers 1, 2, 3, and 4 on the picture. There's still 5, 6, 7, and 8. If you have a question that you would like to ask. All right, here's the other 4 parts of the picture. You'll remember from the video that she talked about ligase. It's another enzyme, ends with ASE. And she did not dream up that ligase is like the glue. I called it that before I even saw that video. But it is. It's the enzyme that helps put fragments together. So it's number 5. So it's this yellow circle here. And you'll see it's right over the spot where 2 segments of DNA are not physically attached to each other yet. Number 6 is pointing to Okazaki fragments. So down here on the bottom strand, there are a bunch of little fragments. Only this one is numbered with a 6, but like I could have called this piece, and that one all Okazaki fragments. Number 7 represents the leading strand. Now, in this picture and in the video, it was the one that was on the top, but it doesn't have to be, okay? I want everyone to be aware, just because both of the pictures show that, it doesn't mean that the top is always the leading strand. The thing that makes one of the strands a leading strand is that it's continuously replicated. So notice here, there are no gaps in this strand. The leading strand always follows the direction of replication. So earlier I said there was this arrow pointing towards the left, and that helicase would always move in that direction. Well, the leading strand will go in that same direction. So whichever strand is being synthesized or made in the same direction as replication, and thus that helicase enzyme, it won't have any fragments and it will be continuously replicated. Now it's always getting replicated in the 5 to 3 direction. That means that where we started will be the 5' end, and where it's ending off will be the 3' end of DNA. It always has to get replicated that way. The lagging strand, because DNA has to be replicated from 5' to 3', has to go the other direction so that these little fragments it's making are going from 5' on one side to 3' on the other. New DNA has to be synthesized in that direction. That's the way the enzyme works. So that means it'll make a little chunk, jump ahead. Make a little chunk, jump ahead. Make a little chunk, jump ahead. Those are the fragments that we call Okazaki fragments, and they get linked together by the ligase enzyme. So that's why I didn't put any definitions up there, because I described it in that